is May 30th, 2019. I'm doing the second part after the 48-hour incubation period for the MRVP broth, where the S. epidermidis and the E. coli microbes were incubated. So here is my work area. This is biochemical testing for microbial identification, methyl red, Voges-Proskauer, and catalase testing. So I have my MVP broth, that is the E. coli, and I've already poured the S. epidermidis, and I'm going to pour the E. coli. So I'm going to flame the lip in one test tube. Part where I'm going to be dropping methyl red into it. I'm going to cut the tip. Oops, that went somewhere. I'm going to wipe the scissors. This is my area that I can wipe it on. I am to drop 7 drops into one of the E. coli and 7 drops into one of the S. epidermis. So here we go. 1, 2, 3, 4, 5, 6, 7. 2, 3, 4, 5, 6, 7. I'm going to put this to the side. Okay, I'm going to immediately observe the color of the broth. Wow, this one's really red. This one's orange on top. So they're both really different. This is the E. coli is orange and the S. epidermidis is red pink. This is positive. This one is positive. The red or pink in color. I do not like this test tube back. Since the S. epidermidis is the positive, the red color, it is an indicator that it has a pH between 4.4 and 6.0, which methyl red turns red in that pH level. So methyl red is used to indicate fermentation of how the acid and is in the different microbes. So it's a mixed acid fermentation test. So the one that turned orange color is an indication that it is not a mixed acid fermentation and it's neutralized. It turns yellow. So that is considered a negative test result. The next one is going to be the Voges-Proskauer test, which is to identify if it ferments glucose. Test tube rack kills me. The next test is the Voges-Proskauer test, which is to identify microbes that ferment glucose. This uses what's called a Barrett's reagent. I have a Barrett's A, and then I have a Barrett's B reagent. So I'm going to cut these tubes and I'm going to add 12 drops of each. Okay, I'm going to remove the tip of Barrett's A reactive reagent and add 12 of each. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. I'm going to swirl it around and mix the other one, swirl that around. Then I'm going to get the Barrett's reagent B and drop 4 drops. Here's 1, 2, 3, 4. 1, 2, 3, 4. As to shake the tube. At this point, I need to shake both tubes for 30 seconds and then let them sit for 30 minutes. So I will be moving on to the catalase testing, and then we will revisit the results afterwards. The next part of the experiment is the catalase testing. I have the 2 slides labeled E. coli and S. epidermidis. I have the 2 culture plates, E. coli and S. epidermidis right here that I'm going to take colonies from. And we're going to be using hydrogen peroxide. The point of this test is to see if the metabolites are reactive and cause damage to cells. So hydrogen peroxide is toxic. That's why we use it on our skin for when we have open sores. And bacteria in the oxygenated environments make the enzyme catalase that neutralizes the harmful effects of the hydrogen peroxide. So when the anaerobic microbes do not have the catalase enzyme. So a positive result will be if the hydrogen peroxide bubbles, which is interesting to me because when you put it on an open sore, it bubbles, and that would mean that it's a catalase positive microorganism that's in our cut or wound. So I find that interesting. So we are going to, and catalase positive means that they are aerobic, not anaerobic. Aerobic means they use air. So here we go. I got my things in alcohol, my loose in alcohol, and I'm going to dry it off, and I'm going to open the lid of the E. coli, take a sample, scraping of what we have. And in the loop, and then I'm gonna pipette 1 drop of hydrogen peroxide on the sample. So I'm gonna put it right here. Cayenne bleach. One drop of hydrogen peroxide. Bubble. There it is, bubbling. So that guy's bubbling, and I'm going to do the same thing to the next one, which is epidermidis. I'm going to gather some colonies. Hydrogen peroxide. Oops, I dropped it on the plate. Let's see. This guy bubbles too. A lot, way more than the E. coli bubbled. This guy's insane bubbling. So that is the catalase video. This is the Voges-Proskauer test, and I'm going to show you the results. The E. coli did not change any color, just got cloudy. And the S. epidermis has a slight pink layer at the top, and that is a positive result for the Voges-Proskauer test, and that means that it ferments glucose. And this test is uses the fermenting glucose through the I can barely say it, butadiene pathway, and that's what giving the Barrett's reagent is added for to produce a weaker acid than the mixed acid fermentation, and the middle part of that would be the butadiene fermentation, which is acetone. And this acetone makes it red at the top. So S. epidermidis is positive with the red at the top. And that ends my video for biochemical testing. Thank you.